All right, hey everyone, these are your protein notes. So I'm going to go ahead and just jump right in. Again, proteins are one of the four major macromolecules that make up living things. Proteins are polymers. Again, poly means many, which means that they're just going to consist of a lot of different individual subunits put together. Those subunits, those monomers, are called amino acids. Again, mono means one. Again, pardon my handwriting. This is kind of funky on the on the computer, but whatever. All right. So these amino acids, kind of like we talked about with carbs, are going to have kind of a dry and a kind of in solution formation. If you have dry amino acid, which means it's just out of water, you're going to have no charges on your amino groups and your carboxyl groups, right? So if you look at the overall architecture, you have a central carbon. Every amino acid has that. You have one hydrogen. You have the amino group. You have the carboxyl group, the COOH, and you have what's called an R group. The R group is the only thing that changes between amino acids, right? If you drop amino acids in water or in solution, like in your cells, here's the charges I told you about. The carboxyl group becomes negatively charged and it loses a hydrogen. The amino group becomes positively charged because it gains a hydrogen. And again, the R group and the hydrogen are still there. They are, well, the hydrogen's always there. The R group is going to be different for every amino acid. So this is your basic architecture. And if you would like to pause, I'm actually going to request that you pause, take out a piece of paper, and draw an amino acid by hand from memory. This should be something that you can do no problem. The basic structure is always the same. You can go ahead and just write R for the R group, but go ahead, do that. And I'm going to continue. All right, linking polypeptides together with carbs and proteins is essentially the same. It's dehydration synthesis, so that word got left off here. Dehydration synthesis, we're going to remove a water when we do this, or I guess when enzymes do this. Also called a condensation reaction. But this reaction happens between adjacent amino acids. We're going to take an oxygen from the carboxyl group of one amino acid and two hydrogens from the amino group of the other adjacent amino acid. And that's where your water molecule comes from. That's where your H2O comes from. And as a result, the carbon of this carboxyl group forms a covalent bond with the nitrogen of the other amino acid's amino group. And that bond is called a peptide bond. That bond is called a peptide bond. And again, this is all enzyme mediated. All of this is essentially going to happen in the ribosome, which is just one giant enzyme if you want to think of it like that. But that's how you link amino acids together, right? Again, synonym for all this is the condensation reaction, and that bond that we just drew is the peptide bond. When we talk about protein synthesis, the direction does matter. The protein grows, or the polypeptides, a synonym for protein, grows in the direction of whatever side the carboxyl group is on. So in this case, we're going to add to the right here. All right, so let's talk about the R groups. This is where kind of the detail is going to really matter, and I want you guys to focus most of your attention. You need to be able to look at an R group, which is this boxed area. It's the part that changes in every amino acid. You need to be able to look at that and infer how it's going to react with other amino acids and how it's going to react when the protein as a whole folds during tertiary folding or secondary folding and secondary structure and tertiary structure, right? So there are five or six things I want you to pay attention to. You should be able to identify if the R group has no charge or if it does have a charge. you should be able to identify if the R group is polar or nonpolar. You should be able to identify if there are any disulfide linkages. So you're going to look for sulfur in that case. I'll say disulfide bonds. And you should be able to identify if that R group is hydrophilic or hydrophobic. Hydrophilic or hydrophobic. So if you want to really quick, pause and try to do this before I go over these examples. Look through all of these different R groups that are boxed and go ahead and figure out if you can identify any of these characteristics. All right, so here we go. I'm assuming you've done that. Let's start with this first one. If you notice, this R group has no OH at all. There's no hydroxyl groups. These carbon-carbon hydrogen bonds are all non-polar covalent, and there's no charge here. So you would say there's no charge in this R group. And this is hydrophobic. I'm not going to write hydro, but I will write phobic. So hydrophobic, just to save time because it's slow for me to write. And that's it. That's it. So let's go ahead and look at some other amino acids. Let's look at threonine. You should have immediately noticed you have a hydroxyl group in threonine, which means you have a polar covalent bond making this R group polar and making this R group hydro, oops, I wrote hydro on accident. I meant to just write philic, making this hydrophilic because of this OH. All right, and asparagine, which I believe shares its name with asparagus. Oops, asparagus. It's not why your urine smells, but I think the name is the same here. You should immediately identify that, hey, this oxygen is charged. So this R group is charged. And because it is charged, it is going to be hydrophilic. Hydrophilic. Let's look at cysteine while we're up here. There's a sulfur and cysteine. So this amino acid's R group is going to want to engage in disulfide linkages. Let's go down here. Glutamic acid has a charge. So this is charged and therefore hydrophilic. And in arginine, this nitrogen right there has a charge. So this is also going to be the same. It's going to be charged and hydrophilic. So I'm not going to write that twice because it applies to both of these. All right. What happens, and this is the key thing, what happens when you switch R groups out? You need to know the effect of this on the overall protein. So what happens if you switch these out, I'll say with each other, right? So basically what I'm asking is if you have a protein and a specific amino acid has a charge on it, and there's a mutation, for instance, that substitutes a different amino acid that maybe like this one is nonpolar, how is that going to affect the overall shape of the protein? These are questions that you're going to have to know the answer to because it's going to be the foundation of everything else we do for a lot of the rest of the year. So let's look at consequences, and we're going to do that visually down here. So I'm going to keep scrolling. And let's look at the overall protein structure. We're going to start with primary, work our way down. Just to kind of get oriented, here's all the individual atoms in a protein. You should be able to pause this video, circle the individual amino acids, and highlight the individual peptide bonds between them. So go ahead, do that really quick. All right, welcome back. So here we go. You've got one amino acid here. Again, here's the amino group. on the left, here's the carboxyl group on the right. Right there in the middle is your central carbon. There's your R group, and there's your hydrogen, right? Basic structure every single time. Here's another one. Here's another one. Here's another one. And there is a fifth one. In between these are the peptide bonds. So this first peptide bond is labeled for you, but there's a peptide bond there. There's a peptide bond there. There's a peptide bond there. So you guys should be able to identify, and there's one there, all four of those peptide bonds. All right, on one end, you're going to have an amino group, and on the other end, you're going to have a carboxyl group. So that's what we're showing you down here. Each one of these little circles is an amino acid, right? Each one of these is an amino acid. So just for simplicity, you're not always going to see all the atoms, you're just going to see these little balls, right? It looks like a necklace, right? And then the lines between them are the peptide bonds. All right, so your basic structure is your primary structure. It is simply the order of amino acids. It's just the order of them. Which amino acids come after which amino acids. Actually, before we continue, let's list some proteins. I forgot to go ahead and do that. So over here on the side, let's just list some proteins, because you guys are familiar with some proteins even if you don't realize it. Take a minute if you want to pause and kind of think about it. If not, I'm going to continue. So here we go. We will talk about actin and myosin. Actin and myosin are muscle proteins. This is why you can move, because these are contracting and helping your muscle fibers contract and relax. Actin and myosin. You might be familiar with keratin in your hair and your nails. You might be familiar with collagen, the protein that makes your skin nice and beautiful when you're young. You might be familiar with hemoglobin in your blood. Carries oxygen, has iron in it. You might be familiar with mouth enzymes, amylase, the digestive enzyme that breaks down carbs in your mouth. This is why breads and stuff taste sweet when you eat them. It's because amylase is working away on those polysaccharides, breaking them down into those sweet monosaccharides. Pepsin, trypsin is another one. Enzymes that focus on digesting proteins, like the peptide bond in proteins, hence this is why it's called pepsin. Whoops, that was my Roomba. So those are some examples. All right, let's continue. So secondary structure. Secondary structures where things start to get a little bit more interesting. Over here on the side, this is due to the hydrogen bonds, not the R groups, only the hydrogen bonds between the carbon and the nitrogen backbone. And I'll say in parentheses, not R groups. We're not looking at the R groups yet, right? So here's what I'm talking about. Here's a carboxyl group from one amino acid, right? Here's an amino group from a different amino acid somewhere else on that chain. The hydrogen from that amino acid's amino group is forming a hydrogen bond with the oxygen from some other amino acid's carboxyl group, right? So what we're really doing is we're forming hydrogen bonds in between this nitrogen carbon backbone that runs right down the middle of this entire, I'm kind of tracing it over right now, this entire protein, right? Those are where those hydrogen bonds form. If the folding looks kind of like this loopy-loop, like on a roller coaster, which is a pretty common one to see, I'll say like, oh, I don't know, half the time you might see this one, that's called an alpha helix. Sometimes the folding is a little bit more kind of rigid and linear, and it kind of looks like you've accordioned a piece of paper, and the hydrogen bonds form that way. That is called a beta pleated sheet. A beta pleated sheet. And again, this is all due to hydrogen bonds between the carbons and nitrogens in that backbone. So not the R groups and not the hydrogens that stick out the top here. Just the carbons and the oxygens. If you do have a hydrogen involved, usually it's from one of the amino groups, like right there, right? Same thing here. Oxygen from a carboxyl group, hydrogen from an amino group, and between those two, they have formed a hydrogen bond. So that's secondary structure. All right, tertiary structure. You should be able to identify all of these interactions in this worm-looking protein drawing. It looks like an earthworm, but it's really just a protein and all the little balls are amino acids, right? So pause, try to find them on your own. All right, so here we go. We're gonna do this one together. I'm gonna put a star by this because this one's gonna be important. All right, we're looking for H bonds. We're looking for hydrogen bonds. So in this case, we're looking for partial positives and partial negatives. So here's a hydrogen bond right here, right? So this partially charged positive hydrogen will be attracted to this partial negative oxygen. That's a hydrogen bond. They also exist as part of tertiary structure. They also exist as part of tertiary structure. By the way, this is where we look at the R groups, right? And the reason why you can have hydrogen bonds in tertiary structure is because, well, some R groups have hydrogen as part of them. Actually, one amino acid has hydrogen as its R group. So they show up here too. Disulfide bridge or disulfide linkage right here in the middle. Ionic bonds, you're looking for a positive or a negative charge, not a partial, a full-on positive or a full-on negative charge. So here's an ionic bond right here. It's actually labeled for you. The oxygen here is negative, and the nitrogen there is positive. You should be able to, let's see, there's an R group right here that I'm circling, adjacent to an R group, actually an identical R group. These two R groups right here, kind of at 12 o'clock in this image, have only nonpolar covalent bonds. All the carbon-carbon, carbon-hydrogen bonds are nonpolar covalent. So these bonds, or these interactions right here, are going to be hydrophobic. I'll say H-phobic. Hydrophobic. And what happens, and this is why we're talking about folding, is that R groups that are hydrophobic will tend to want to kind of clump up and essentially hide from water, because if you have a protein in the cytoplasm, that's watery cytoplasm. So your hydrophobic R groups are going to want to hide together, and that's going to influence the folding of your protein, right? Let's see, hydrophilic interactions, anytime you have a charge. So again, your ionic bonds, you're going to have a hydrophilic interaction there potentially. Actually, sorry, I'm incorrect. Water is going to want to break apart this ionic bond. So the ionic bond is actually going to want to be inside, kind of towards the middle of the protein. Your hydrophilic areas are going to be more along the lines of your partial positive and partial negative charges. Right, so this area would be hydrophilic. Any R group that's hydrophilic is going to be more likely to stick outward, because that's where most of the water is going to be. R groups that are more hydrophobic are going to want to fold up and be towards the inner part of this protein where there's less water. Right, so these hydrophilic and hydrophobic are considered just to be general interactions. But you should be able to see that if you replace R groups, you're going to change the fundamental folding of the protein. And that is the take-home point here, That's the take-home point, because all of these R groups which affect folding affect the final functional shape. If you alter the shape at this point, that protein is either going to not function as well or not function at all for the most part. So this is the final functional shape for your proteins. I cannot emphasize the importance of protein folding enough. I'll say it over and over and over and over again. So over here, this is just a picture of a protein in its tertiary structure. Notice they're not going to show you the primary structure because that's way too detailed, but you can see elements of the secondary structure. You can see, for instance, the beta-pleated sheets and the alpha helices. And depending on the protein you're looking at, sometimes you'll have both. Sometimes you might only have beta-pleated sheets. Sometimes it's only alpha helices. But each protein is unique into how many of those it has. But every type of protein will look the same. Like all keratins will look the same. All collagen proteins will all have the same folding, right? They'll all be the same because they're the same protein. All right, last little bit here. Quaternary structure. Some proteins need quaternary structure, some don't. So I'll say some proteins require this or need it. Two examples, again, are collagen, makes you look nice and young and beautiful, is actually a combination of 3 separate proteins that kind of interact and kind of look like a rope. Hemoglobin actually has 4 different proteins. It has what are called alpha chains. This is different than alpha helices, so just actually kind of ignore that for right now. And actually 2 of them are what are called beta chains. And again, those are a little bit different than beta-pleated sheets. They're not the same thing, so don't get too caught up on that. It actually has iron in it, carries oxygen, right? So these are some examples of proteins that have a little bit more complex structure than just a single protein. Let's take a little bit of this space over here on the right and just do a quick summary. So I'll cordon this off and we'll summarize. So primary structure is the sequence of amino acids. So I'll say amino acid, AA, order, including the peptide bonds that hold them together. All right, secondary structure are the hydrogen bonds in the carbon-nitrogen backbone. Tertiary structure are group interactions. R group interactions. And then quaternary structure is just multiple proteins interacting with their R groups to make a larger functional protein. All right, so let's draw a line right here. How do you break these things? How do you break a protein? That's a question mark. You need to know this. So in other words, how do you, denature, that's the fancy word for breaking a protein. How do you denature a protein? And you do need to know this. I'll star it. There are 3 ways. Take a quick second, pause the video, brainstorm. All right, let's see if you got these 3 ways. Here we go. Way number 1 is with temperature. Kinetic energy. It can break apart the covalent bonds, the hydrogen bonds, mostly, again, the hydrogen bonds, but if it's hot enough, it can break really any bond and denature that protein. So high temperature, specifically it's high temperature. Low temperature just tends to slow the molecule down. It doesn't really break anything. It's in particular, it's high temperature, high kinetic energy. The second one I have is pH. The concentration of hydrogens or hydroxide ions. I'll do the H first, OH. The third is salinity. This is probably the least known of the three. Salinity. The mechanism for salinity is a little funky, so don't worry about the mechanism behind it. pH can affect proteins similar to temperature. It doesn't denature it using kinetic energy, but the hydrogen ions and the hydroxide ions, or the hydroxides, they will surround either a negative or positive part of the protein and kind of force it away from the rest of the protein and kind of snap it off and break it off. So the end result for pH is the same as it would be for temperature. All right, one thing I want you to know, so the last thing here, it's good to commit this to memory. The salinity of pure water is 0%. The salinity of your cells, so I'm going to put IV, like when they give you it in a hospital, they give you an IV. They match the salinity of your cells so your cells don't undergo too much osmosis, take in extra water, and burst. So the salinity of a hospital IV is the same as the salinity of your cells. I'm going to leave that blank. I want you to think about that while we talk about this. What do you think that percent is? And we're going to compare all of this to the salinity of ocean water. And I'll give you a number here. Ocean water is about 3.5% salinity, salt. So if you thought we were like 10% here, you're way off, because we're less salty than ocean water. We are actually 0.9%. 0.9% is the salinity of your cytoplasm and of your blood. Right? So this is us. This is us. There we are. All right. Kept it under 30 minutes. Hope this helped. See you guys for the next video.